Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties in the course cell and molecular biology. So far what we have discussed about the evolution, we have discussed about the origin of life, and subsequent to that we have also discussed about the different cellular structures of the prokaryotic as well as the eukaryotic cells. In that context, we have discussed about the prokaryotic cell, where we have discussed about the cell wall and other kinds of properties. Whereas in the case of eukaryotes, we have discussed about the different cellular structures. We have discussed about the different types of organelles, and we have also discussed about the function of these organelles. Now, in today's lecture, we are going to discuss some more aspects related to cell and molecular biology. So, when we talk about the living organisms, right, where the basic feature of any living organism is that it is actually going to have the complex organizations. It should be responsive to the different types of environmental stimuli, such as temperature, wind, starvation, and so on. But apart from that, the fundamental basic property of any living organism is that it should be able to give rise to the offsprings. That means you should be able to reproduce, and on the other hand, it should be able to have the endogenous growth. So these are the two properties which actually discriminate a living organisms from a non-living organisms. And if you recall, we have discussed about the different types of cells. We have discussed about the prokaryotic cell, we have discussed about the eukaryotic cell. Within the eukaryotic cell, we have discussed about the animal cells, we have discussed about the plant cells, we have discussed about the yeast and as well as the fungi. But the basic phenomena of any living organism is that it wants to contribute into two different types of fundamental activities. One, it wants to increase its number, and it also wants to grow in size. I am sure you might have seen that when you are born with a small baby, right? And in a due course of time, you have taken the nutrition from outside, and that's how you are actually being developed into adult individuals. Same is true for the even the tiny cells. So if you see the cell is actually going to perform the different types of activities, and these cells are mostly would like to do all these activities so that they can be able to grow in size, and they can be able to make more of their type, or they will be able to divide and give you the different types of similar kind of cells. So the growth is the fundamental basic property of any living organisms. So growth is the fundamental nature of an organism, right? Growth is the outcome of the cell division, which means one cell is going to divide into two, and that's how it is actually going to have the growth in the number. But all these tiny cells are also going to grow in size in due course of time. The cell division is a process in which each parental cell reproduces by dividing into the two daughter cells under the specific conditions. The resulting daughter cells can themselves grow and divide, and giving rise to the new cell population formed by the growth and the division of the single cell. In this context, I am sure you might have studied when you were doing the microbiology experiments in your laboratory, that when you are going to inoculate a single tiny bacteria, right, it actually grows into, it divides and then divides and give you rise to the complete cell culture, right. Similarly, if you plate these few bacteria, you will see that they are actually going to form the colonies. These colonies are actually containing the different thousands of bacteria and so on. So in a simpler format, the continuous cycling of the growth and cell division allows the single bacterium to form the colony consisting millions of progeny bacteria following the overnight incubation on the nutrient agar media. And this is what you might have done in your microbiology labs. In a more complex form, when you are going to do with the eukaryotic cells, the eukaryotic cells are actually going to be developed as a single cell. So for example, in the humans, when the sperm and ovum are going to fuse, they are actually going to give the single cell individual, and that is being called as the egg or the zygote. And then these zygotes are then going to go with the repeated divisions, right? And that's how these divisions are give rise to the 10 to power 14 cells, and that's how it is actually going to give rise to the small baby. And then that baby is also going to take up the nutrition from outside, and that's how it is actually going to grow up into an adult individual. So the division of the cells must be tightly regulated and coordinated with the cell growth and the DNA replication to ensure that the proper flow of genetic information should be maintained, right? So this is the most important part what we are going to discuss in this particular module, that the cell is going to divide. So we are actually going to discuss about the division mechanisms, how the different types of cells are utilizing the different types of division mechanisms. On the other hand, it is also very important that all these division mechanisms should be tightly regulated by the cellular machinery, and it should be under coordination so that it should not give you the the irregular cells, irregular multiplications or irregular divisions. And we are also going to discuss what will happen if any such thing actually going to happen. Now the fundamental question is why we need to grow, right? Why the growth is required. So for that I will take you to the one of the examples. Suppose you are cutting the vegetables in your kitchen, right? Or suppose you are doing the any kind of paper cutting and all that, and all of a sudden then it actually you cut your finger, right? So what happens when you cut your fingers? When you cut your fingers, the blood is started oozing out. And why it oozes out? Because you are actually cutting the blood vessels, and blood vessels are made up of the specialized cells. These are called as the endothelial cells. But it doesn't happen that you are actually keep losing the blood, right? In due course of time, what happens is that the body is getting the signal from the cutting site, and that's how you are actually going to start doing the tissue repairing and all those kind of things. And the tissue repairing is requiring that the neighboring tissue which are still healthy and there does not get any kind of injury is going to grow, and that's how it is actually going to pile up or complete the tissue site. Okay, so when you get a cut or when you get injured, right, the body is actually the surrounding tissue of the injury undergoes the inflammation. So inflammation is a protective immune activity, right? This anyway we are going to discuss when we are going to discuss about the basic immunologies. And it is actually going to release the many type of growth factors. It is also going to release many types of chemoattractants so that the neighboring tissue or neighboring cells from the neighboring should come and protect this particular cut site. And then it's also going to induce the different types of signaling pathway, and that's how it is actually going to induce the cell division. Because if you are getting a cut here, right, suppose you are getting a cut here, the neighboring tissue is actually going to start developing. I'm sure you might have seen when you are getting an injury in your hand or feet or whatever, right, it is actually forming a clot, right? And that's how it is actually going to, all the cells are actually covering this wound site, and then in due course of time they get completely healed, and then this portion is going to be peeled off, and that's how you are actually going to have the recovery of the injury. So your body is made up of the billions of cells, and those cells make up the multiple copies of cells to fix the injured tissue so that you can be able to function properly. Because this is very important, because if you don't get the recovery of your injury, then it is actually going to damage the cell machinery. The average human body contains 10 to power 14 cells, and these cells contains more than 200 differentiated cell types. The cell division is crucial to replace the cells that have been died by the senescence or the necrosis for the tissue maintenance. So this is also one of the portion what we are also going to discuss at the end of this particular course, where we are actually going to discuss about the what happens when the cell is not getting the enough nutrition, and how the cell will actually go through with the death pathways. Now, as I said, the cell division is a controlled event, right? So cell division is a controlled event because you have to divide the cells in such a way so that it should not give you the enough amount of cells. So in high eukaryotes, the progression to the different stages of the cell cycle is controlled by a series of protein kinases which have been conserved from the simple yeast to the highly complex animal cells. And this happens even in bacteria also, and this happens in the higher eukaryotes. The cellular machinery is itself tightly regulated by the growth factors that control proliferation, and it allows the cell division events to occur only in the when the organism is required. So this is very important also. And the defects in the regulation of the cell cycle causes by the genetic instability are common cause of the abnormal proliferation of the cancer cells. And this is very important what we should understand, that if there is a dysregulated cell division which may actually not under the tight control, then these cells are only going to differentiate in due course of time, and that's how they are actually going to develop the cancer cells. So cancer is one of the leading cause of death, claiming millions of lives every year across the world. So first we have to understand how the cell is going through the different types of cell division or the different stages of the cell division, so that it will be understood that at every stage there is a way in which you can be able to control the cell cycle. So, cell cycle, the self reproduction is the most common characteristic of the cell, right? Every cell wants to grow, everyone want to make its own copy so that it should remain into the environment. All cells reproduce when the parental cell divide into the two daughter cells, and after every successful completion of the cell cycle. The cell cycle of the most of the cell consists of the four well coordinated process. One is cell growth, then the second is division replications. Then we have the segregation of the replicated chromosomes, and then we have the cell division, which means there will be a division of the grown cells. So the cell growth is actually been also called as G1 stage. DNA replication is going to occur into the S phase. Then segregation of the replicated phase is going to happen in the M phase. So after the M phase, the cell will enter into the cell division, and that's how the one large cell is going to divide into the two daughter cells. So in bacteria, the cell growth and DNA replication occur through the most of the cell cycle, and the duplicated chromosome distribute to cell division in association of the plasma membrane. In high eukaryotes, the cell cycle is more complex and consists of four discrete phases where the chromosomes are duplicated in one phase and the duplicated chromosome distributed into the daughter nuclei before continuing the cell division, right? So what we have said is actually the basic fundamental aspect what is going to happen, whether it is a prokaryotic cell or the eukaryotic cell. So you're going to have the cell growth, which means the cell, suppose this is the normal cell. First, it is actually going to synthesize the cytosol, so that it's actually going to have the large cell, right? There will be a cell growth. And then suppose this is this has contained one nuclei, then this cell is actually going to contain two nuclei, right? And that is what is going to happen in the DNA replications. Then these two nuclei are actually going to be segregated in the third step, and then there will be a cell division, so it is actually going to be divided into the half, and that's how you are actually going to have the two daughter cells, one which contains this nuclei, the other one which contains this nuclei. And this is what is the fundamental events what is going to happen, irrespective of whether it is a prokaryotic cell or the eukaryotic cell. In prokaryotic cells, since the machinery is small, the cellular machinery is also important, small, and the protein machinery is also small. The number of components what required for controlling these cell division or the cell cycle is small. But in the case of eukaryotes, all these are actually going to be under tight control, and that's how they are actually going to be forming these kind of different stages, where you have the G1, then you have S, and after the M phase you are actually going to have the division of the cells. Now this cell cycle and the discovery of the cell cycle doesn't stay in the single day, right? People have done a lot of experiments and lot of discoveries so that you can be able to understand the cell division. It started with the simple discovery of the compound microscope, because when you discover the cells, you discover the cell because there is a compound microscope, and that is being done by the Janssen and Janssen, right, in the year of 1590. So they have discovered the first compound microscope, and because of they have discovered the compound microscope, you can be able to see the cells, right? You cannot be able to see the single cell before that, right? Then year of 1661, the Italian microscientist Marcello Marpelli was among the first to use a microscope to describe the thin slice of the human tissue. So this is the scientist who first saw the animal tissue. Then year of 1665, the Robert Hooke, who coined the term cell after observing the thin slide of dried cork under the compound microscope. And you know that he has seen the dead cells because he has seen the cells were like forming the hexagonal compartments, and that's how these compartments are being called as cells, right? And what he has seen is the cork, the dead and dried cork, right? Then year of 1675, Antonio von Leeuwenhoek was the first to observe the living, free living cells. Then the 1824, the René Dietrich correctly concluded all the animal cell and plant cell as the aggregate of the globular cells. Then year of 1838, the Schleiden and Schwann actually calculated the cell theory. And in the year of 1852, Virchow published the classical textbook which is called as Cellular Pathology, and it is correctly used that the functional unit of life is the cell, where the primary site of disease and the cancer. And the year of 1953, Watson and Crick actually discovered the structure of the DNA, and that's how people have started understanding the cell and molecular biology of the different organisms and what are the different types of events which are being regulated. So one of the major important thing which is related to context to the this particular module is we should understand what is the cell theory, and because cell theory is being proposed by the Schleiden and Schwann, and according to the cell theory, so cell theory was proposed by the German scientists Theodor Schwann and the Schleiden in the year of 1838, and it explains the basic nature of all the living organisms. What it says is, number one, living organisms are made up of the cell, right? So that is very fundamental informations. What it means is that cells are the smallest unit of life. Each cell contains the cytosol with the phospholipid bilayer, right? And all the cells contain the DNA, which holds the information necessary to produce the molecules for the survival, right? So the Schleiden and Schwann postulated or given the cell theory saying that all the living organisms are made up of these cells, and these cells are actually containing the cytosol, right? So these cells are actually containing the cytosol, which is covered or which is actually having a plasma membrane outside, right? So this is actually going to have a covering of the lipid and the proteins, and then it also going to have the DNA, so that DNA is actually going to have all the information required for controlling all the activities of this particular cell. Then the cells are the basic and structural and functional unit of the life. So organism can have single cell, or it can carry all the necessary molecules for the metabolism, or there can be complex tissue which contain different cells to perform the specific task in collective. So group of tissue contains which can actually going to form the organs. So irrespective of whether the organism is unicellular or the multicellular or the organ or the tissue specific organizations, what it says is that the cell is going to be the basic structural and functional unit of the life, which means you can actually have the different types of organization. You can have the cellular level organizations, you can have the tissue level organizations, you can have the organ level organizations, or you can have the organ system organizations. Irrespective of the organizations, the basic function what is going to be performed, it is going to be performed by the single cell, and the cell is the structural and functional unit of the life. Then all cells are derived from the pre-existing cell, which means all the cells are actually going to divide, and that's how they are actually going to form their own copies, and these cells are actually going to perform the different types of functions. So all cells are arriving from the pre-existing cells only. Only the cell grow in size and reach to a threshold, then it undergoes the cell division, giving rise to the two daughter cells. So this is the photograph of the Schleiden and Schwann, right? And so this cell theory was very important, and it actually gives the very important information about that the living organisms are made up of these cells. Cells are the structural and functional unit of the life, and the every cell is coming from the pre-existing cells, and that actually gives the idea that there is a cell division which is very important. So now first question is the cell division in prokaryotes and the eukaryotes are same, right? So what you see here is that the generation time of the different organisms. So what you see here is the E. coli, Bacillus subtilis, Mycobacterium tuberculosis, Eugenia, Giardia and Saccharomyces cerevisiae. So this is actually the eukaryotic organisms, and these are actually the prokaryotic organisms. And what you see here is that the doubling time for these organisms are much faster or much smaller, actually I will say, compared to the eukaryotic organisms, right? So eukaryotic organisms are taking 2 hours, 18 hours and more than 10 hours, or they are working in the frame of hours, whereas the prokaryotic organisms are actually working in the frame of minutes. So basically the cell division and the fundamental machinery or the fundamental process remains the same between the prokaryote or the eukaryotic cells, but the mechanisms, the players are different. So although the basic events occur during cell divisions are similar in both the prokaryotes and the eukaryotes, the cell division is more complex and highly regulated into the eukaryotes, and because of that only the prokaryotes are actually having the shorter lifespans or the shorter generation time, whereas the eukaryotes are actually having the more generation time. And that is understood because you have to perform a lot of functions, right? And you will see in due course of time that why it is bigger in eukaryotes versus why it is smaller in prokaryotes. Eukaryotes have several checkpoints where the cell regulation is progression in cell cycle, and each checkpoint of the cell cycle has to be cleared to progress further into the cell cycle, else there will be a cell cycle arrest at that particular point. The multiple checkpoint increases the time required to complete one cell division. You can observe this by simple tiny cells to take the complete one cell division. A simple bacteria E. coli takes only 20 minutes to complete its cell division, whereas the budding yeast only takes 90 minutes and human cells actually takes more than 24 hours. As the cell complexity increases, the time taken by the cell to divide also increases, and this is what is very clear from this particular table, that if you are a prokaryotic cell, you are going to be take the smaller time to divide. If you are the eukaryotic cells, then it is actually going to take more. And why the prokaryotics are dividing so fast? So cell cycle in prokaryotes actually is taking lesser steps and dividing. So why the prokaryotes are dividing fast? Because the prokaryotes have the simpler cell cycle and most commonly the binary fissions. First, the cell grow in size by accumulating the nutrition under the favorable environment and condition, and once they reach the optimal size, they initiate the chromosomal duplications. Chromosomal replication is a very crucial process in cell division, and it must be tightly regulated. Once the chromosome is replication accomplished, the molecular mechanism must ensure the successful segregation of the daughter chromosome into the daughter cells, and prokaryotes tightly regulate their chromosome replication, and this is what we are going to discuss, right? So how the prokaryotes are actually regulating the distribution of the chromosomal replication. Remember that the prokaryotes does not have any kind of partitions, they don't have any kind of nuclear membranes and so on, right? So within the single cell you actually have the DNA, right, which has been part of their genome, right? So this genome is going to be duplicated, and then this duplicated genome is actually going to be distributed between the within the cell, so that when there will be a cell division, it is going to be equally divided. So how the regulation of the chromosomal replications? Remember that when we are going to discuss about the replications, you will understand that there are different mode of replications, but the most acceptable mode of the DNA replication is the rolling circle model, right? So in the rolling circle model, you are actually going to use the one point, and which is being called as the origin of replications. So in the bacterial chromosome, the replication of the chromosome starts at a specific sequence which is called as the origin of replications. So original replication is the site from where the DNA is going to start replicating, and then it is actually going to form the mechanism which is being described for the rolling circle model. So when we will discuss about the replication in the prokaryotes, that time you will understand the more detail about the rolling circle model. A replication for factor which is called as DNA binds to the specific sequence within the origin C and then initiates the unbinding of the DNA and the loading of the replisomes. DNA is regulated by the ATP binding, and it becomes active when the ATP is linked to it. Other regulatory mechanism which inactivates the DNA includes the competitive binding at the ODC, repression of the DNA, titration of DNA ATP away from the chromosome, and the inactivation of the DNA ATP. So all these mechanisms are very important, and you see all these are linked to the ATP, right? So the molecule which are central to this whole activity is ATP, and ATP is linked to energy, right? So if the bacteria does not have enough amount of energy, and energy is linked to the nutrition, right? So if there is not enough quantity of nutrition, it will not be able to produce the enough quantity of ATP, and if it cannot have the enough amount of ATP, then this whole event is actually going to be slowed down, right? Because the DNA is not going to bind these specific sequences onto the ODC, because it requires the ATP and other kinds of molecules, and that is all linked with the energy, and energy is linked with the nutrition. Before the initiation of the replication, both the strands of chromosomes are methylated at the adenine with the sequence of GATC. Immediately after replications, only the parent strand do have the methyl group, and hence called as hemimethylated DNA, which facilitate a competitive binding between the DNA and the seq-A. Hemimethylated DNA is strongly bound by the seq-A. Binding with seq-A at ODC blocks the DNA binding and hence prevent reinitiation of the chromosome replications. Then another regulatory mechanism, so you have what we have seen, one mechanism where the DNA and ATP interactions are very crucial, and the availability of ATP itself is a very important. The other is where you are actually going to have the decrease in the ratio of DNA ATP to DNA ADP. The conversion of DNA ATP to the DNA ADP is catalyzed by the ATPase enzyme called HDAA, and which binds to the DNA close to the replicosome and specifically target the DNA ATP. This is also related to the energy, right? Remember that energy is very important when it comes to the prokaryotic system, because energy is linked to the nutritional status of the environment, right? Nutritional status. And if the bacteria is under the less nutritional status, or the environmental factors are not very conducive, then all these events will go into a state where the replication is going to be slowed down, or the bacteria will increase its generation time and so on, and that's how the bacteria is actually having the immense power of threshold the different types of adverse conditions. But genome replication is fast in bacteria like in the Yersinia pestis or the E. coli. Why it is fast? Because the genome size itself is very small compared to the eukaryotic cells. The circular nature of the chromosome facilitates the faster replication, as the DNA replication occurs bidirectionally from the original replication, and the formation of multiple pore decrease the further requirements of the. So you know that if you have a circular object, right, then you are actually going to start from the original applications. So one fork will go in this direction, the other fork will go in this direction, and that is what the mechanism what we are going to discuss in the when we are going to discuss about the replication into the subsequent modules. And because they go in the different directions, the replication is faster, and apart from that, the DNA size itself is also very small. So once you have the two copies of the chromosome within the bacterial cell, then it is actually going to segregate, which means it is actually going to distribute it to this particular parent cell, so that ultimately it will go through with the cell division, and that's how you are actually going to have the two copies of the bacteria, right? So how we are going to have the segregations? So during the cell division, the segregation of the chromosome to the pole of the cell ensure that each daughter cell get a copy of genome, and it also facilitate the septum formation. Most of the bacteria uses a mechanism which is called as the PAR system to distribute the chromosome and its plasmid equally into the progeny cells. This system is composed of the protein which is called as PAR ATPase, PAR B chromosome binding proteins, and the POP-Z complex, along with the chromosome like the PAR S sequences located near the original replications. PAR system does not segregate the fully replicated chromosome. Instead, POP-Z localizes to the old pole of the cell and facilitate the anchoring of the chromosome to this location by the interacting with the PAR B, which itself bound to the PAR S sequences. Once the replication begins, the newly synthesized PAR sequences bind to the another PAR B, which then pull to the pole of the new cell by the ATPase activity of the PAR A. And POP-Z not only anchor the parental chromosome at the old pal, but also recruit PAR A on PAR B bound on the newly synthesized PAR S sequences. This means what it says is that they are actually going to use a PAR system where the PAR proteins, one protein is actually going to bind the older chromosome, and the other proteins are actually going to bind the newly replicated chromosomes, and that's how at one end the parental DNA is going to remain, and the other side you are actually going to have the newly synthesized DNA. Then you are actually going to have the cell division, right? And cell division is going to be done by the binary fission, right? So several proteins play a role in cell division. Collectively, they are known as the FSTs protein, and they key one of them is FITZ, right? Similar to the tubulin of the eukaryotes, and which play important role into the binary fission. FITZ proteins interact with each other to form a division apparatus called as the divisionosome. So this is a segregation of the chromosomes, right? The chromosomes are segregated to the two poles of the cell. Then in the rod-shaped bacteria, the divisionosome formation begins with the arrangement of the FITZ protein in a ring shape around the center of the cell, which then act as a central division plane. The FITZ binds to the other divisionosome protein, including the FITZ and ZIP-A. ZIP-A act as an anchor which stabilizes the FITZ ring onto the cytoplasmic membrane. FITZ also involved in recruiting the FITZ-Z, another chromosomal protein. Another group of protein known as the min protein, including the min C, min D, and the min E, interact to help in the positioning of FITZ ring into the center of the cell. As cell elongation continue and receptor formation begins, the two copies of the chromosomes are pulled apart each of the tetra cells. So this is what exactly going to happen here, right? So in the middle of the cell, you are actually going to have this cell division FITZ proteins which are going to aggregate, right, or which are going to localize, and then they are actually going to have the other cell proteins which are called as min C, min D, and min E, and then they are actually going to form the elongation of this particular cell. As the cell constricts, the FITZ brings depolymerization, which triggers the inward growth of the cell wall material to form the septum and seal off the daughter cell from others. So this is all about the cell division within the prokaryotic system. What we have discussed that whether it is a prokaryotic system or the eukaryotic system, you are actually going to have the four distinct phases. One, the cell growth, where the cell is actually going to grow into the size. So cell is actually going to grow in size. So if suppose the cell is like this, it is actually going to start synthesizing the cytosol, and so all the cytosol protein are actually going to synthesize, and that's how it is actually going to increase in size, right? And apart from that, the chromosome which is present here is also going to be duplicated. So in the second step, so then the first step, there will be a synthesis of cytosol. In the second step, there will be a synthesis of DNA. So you have the one copy of chromosome, right? and that one copy is going to be duplicated, and that's how the each portion of the cell is actually going to have the two cells, and then it will go through with the division. So this is the cell division, and that's how you are actually going to have the two cells which are actually going to contain their individual chromosomes, right? And it is under very tight control. You have the control by the ATP level. You are actually going to have the control with the help of the different types of protein which are been participating into these events. But these events are the molecular players regulating the cell cycle within the prokaryotic system, and the events are also very fast. Like for example, the chromosome size is very small, and then you are actually going to have the multiple multiple forks which are actually going to run within the genome, and that's how you are actually going to have the faster replication, and that's how we have seen that the generation time in the prokaryotes are very fast. Now let's move on to the eukaryotic system and see how the eukaryotic system is performing the similar kind of events and performing the cell divisions. So in the eukaryotes, the phases of the cell cycle is that cell cycle is actually broadly been divided into the two events. One is called as the interphase, the other one is called as the metaphase or the M phase. Within the interphase, you have the G1 or the G0 phase, then you have the S phase, and then you have the G2 phase. And within the M phase, you are going to have the mitosis or the meiosis, right? And these are the specific type of cell division which we are going to discuss in due course of time. And then you also going to have the cytokinesis, so that it is actually going to divide the cells. So the interphase is actually the phase which is actually going to preparing the cells for the M phase, and the M phase is which is actually going to be used for dividing the cells, right? So let's first discuss about the interphase. So phases of the cell cycle. So a eukaryotic cell divide into two phases. One is called interphase, the other one is called as the M phase. During the cell cycle, the cell spends approximately 90%, 95% of the time in the interphase, and only for 1 hour in the mitosis. The interphase can be divided into the discrete phases like the G1, S, G2 followed by the M phase. G0 phase, so within the G1 you also have the G0 phase. The cells are metabolically active but are not committed to the device. One of the classical example is the neurons, right? Or the RBC, right? So these are the cells which are actually metabolically very active, but they are in the G0 phase. Then we have the G1 phase. So proteins involved in the DNA replications are expressed and the cell prepare for the DNA replication. So within the G1 phase, you are actually going to have the synthesis of the cytosol. And within the cytosol, you are actually going to acquire the machinery which is required for the DNA replication. So you are going to have the synthesis of the DNA polymerases, you have going to have a synthesis of the nucleotides and so on. And then this machinery is going to be utilized into the S phase or the synthesis phase. So DNA replication commences and the chromosomal number is going to be double. So within the S phase, you are actually going to have the duplication of the DNA. Then you have the G2 phase, or I will say gap 2. The cell continue to grow and expresses the protein which are involved into the mitosis. So remember that the G1 phase is actually being required for the synthesis, for the preparation of the S phase, whereas the G2 is required for the preparation of the M phase. Then mitosis is the most dramatic event of the cell cycle, where separation of the daughter chromosome take place, and at the end of the M phase you are going to have the cytokinesis. So this is what it is being shown, right? You are actually going to have the G0 phase, you are actually going to have the G1 phase, then S phase, then G2 phase, and then M phase, and within the M phase you are going to have the cytokinesis. How does the cell cycle begins? So let's understand this by using a model organism such as Saccharomyces cerevisiae. So these are the simplest unicellular organisms or unicellular eukaryotes, where you can actually be able to understand the different types of diseases or different types of processes what are occurring within the cell cycle. So there are various regulatory points into the cell cycle driven by the extracellular signal onto the environment as well as the internal signal that monitor and coordinate the functions. In S. cerevisiae, the first regulatory point occurs into the late G1 phase and it is known as the start. Passing through the start is highly regulated by the availability of nutrients into the environment, mating factors and the size of the cells. Hence, the start acts as a checkpoint where the cell checks for the availability of the nutrients and the required to the complete set of the stages of the cell cycle. Once it proceeds from the start and enter into the S phase, it will become committed to undergo one cell division. So there is a checkpoint before the cell will enter into the into the cell division, and that is called as the start site or start checkpoints. And at the start checkpoint, the cell is actually going to analyze whether there is a proper nutrition available, whether there is a environment which is conducive for the division or not, right? And you remember that these things are very important for a unicellular organism, right? If the environment is not very conducive or environment is not good, like for example temperature is very high, there is a loss of water and so on, then it will decide that okay, let's conserve these nutritions and then let's conserve and then it will go through in those conservative modes. And these conservative modes also we are going to discuss when the cell will go through or the cell will experience these kind of harsh conditions at the end of this particular module. Budding in the yeast produces a progeny cell of the different size, a large mother cell and a small daughter cell. So that is what you see here is a large mother cell and a small daughter cells. The smaller daughter cell must grow into the greater and the mother cell in size before it divides. The start mechanism allowed the cells to reach a minimum size before it can divide. Now, if you talk about the cell cycle regulatory points, so you have the regulations by the one of the protein complexes which are called as CDKs and cyclins. Then you have the regulation by the DNA damage, and then you also have the regulation by the different types of growth factors. And within the DNA damage, you can have the different types of proteins. So you have the ATM and ATRs, you have a P53, and you have the RB proteins. So let's first talk about the regulation by the CDK and cyclins. So remember that the CDK and cyclins are pair of proteins which are actually participating into the regulation throughout the cell cycle. What is cyclin and the CDK? So cyclins and CDK are the two pairs of the proteins which are working together to regulate the cell cycle. So cyclin is a protein which binds the CDKs, right, and causing a major conformational changes which render it in an active form. So once the CDK is going to bind the cyclin, it will turn into active protein, and CDK is known as the cyclin dependent kinase, right? So CDK's full form is cyclin dependent protein kinase, right? So once the CDK is going to be active, it is actually going to start catalyzing the protein phosphorylation, and that's how it is actually going to initiate the different types of cascades of reactions. So CDK is a protein kinase which after getting activated phosphorylate the different target proteins. About the start protein in the yeast, CDK1 which regulates the progression to the start and also enter into the mitosis. Although the cyclin involved in both the regulatory points are different. So if you see the production of the cyclins, what you see here is there is a the level of cyclin with the different types of the stages within the cell cycle. So what you see here is when the cell is under the interphase and the mitosis, or from the so this is the complete cell cycle, right? This is the complete cell cycle. First cycle, this is the second cycle, right? So when it is in the interphase and the mitosis, there is always increase in the level of cyclin and the corresponding CDKs, and that's how it is actually going to regulate the different events of the cell cycle. So you have the CDK1 which is in association with the G1 cyclin, and that's what actually going to bind the cyclin 1, cyclin 2, cyclin 3, and that's how it is actually going to allow the cells to pass the start site. Okay, and once it will start the pass the start site, it is actually going to be committed for the cell division. So CDK1 then associated with the different B cyclins, that is the CLB5 and CLB6, to regulate the progression to the S phase. And then if the G2 to M transition is driven by the CDK1 complex with the mitotic cyclins such as CLB1, CLB2, CLB3 and CLB4, and that's how you see that the CDK cyclin complexes are actually regulating the different phases of the cell cycle, starting from the interface to the G1 phase and then S phase and then G2 phase. In the animal cells, the CDKs are forming the different types of functions, like the CDK4 cyclin D complex is responsible for the progression to the restriction site, the restriction site to the G1. Then you have a CDK2 cyclin E required for the transition to the G1 to S phase. And then you have a CDK2 cyclin A, which functions in the progression of cycle to the S phase, and then you have a CDK1 complex with cyclin A to progress to the S to the G2 phase, and then complex with the cyclin B to regulate from the G2 to M. So this is what exactly is showing here, that you have the CDK4 when it is making a complex with the cyclin D, then it is actually allowing the passing through the cells from the restriction point. If the CDK2 is making a complex with cyclin E, then it is actually allowing the passing through with the G1 to S checkpoints, right? So this is the checkpoints actually you have, and then it will enter into the S phase. Then you have the CDK2 and the cyclin A, which will allow the cell to go through with the S phase, and then you have a CDK1 and cyclin A, which is actually going to allow the cell to pass to the G2 phase, and then it will actually enter into the G2 to M phase, right? And then you have a CDK1 cyclin B, which is actually going to allow to regulate to the G2M phase. So this is actually the different types of CDK and cyclins which are going to be essential for regulating the eukaryotic cell cycle, whether it is a single cell organism such as yeast or a multicellular higher eukaryotes. Then CDKs are actually also having its own regulatory mechanism, right? So once the CDKs are going to bind their cyclins, they will be activated, right? So they will actually going to start phosphorylating the different types of proteins, but they are also going to have the checkpoints, right? They are also going to have other points, other proteins which are actually going to regulate their own activity. So CDK1 has the 3 phosphorylation site, that is the threonine 161, tyrosine 15 and the threonine 14. So phosphorylation at threonine 161 actually keep the CDK1 in the active form. Phosphorylation at tyrosine 15 and threonine 14 by the protein kinase V1 inactivate the CDK1, leads the accumulation of the cyclin 1 CD complex into the cell. And when the cell is ready for the transition from the G2 to M phase, the protein phosphatases such as CDK50 remove the phosphate group from the tyrosine 15 and threonine 14, which then form the actin cyclin CDK1 cyclin complexes. So basically the CDK, when it forms the complex with the cyclin, it gets into the active form, right? And that's how it is started phosphorylating the downstream substrates. But when the cyclin and within the CDK you have the 3 sites. One, you have the T161, you have the Y15, and you also have the T14, right? So this one is actually a site which is required for activation, right? So if there will be a phosphorylation of this site, it is going to get activated. But if the phosphorylation is occurring to this site, then it is actually going to have the deactivation effect, right? So this is what exactly going to happen, right? So CDK, you have the 3 sites. So one is 161, the other one is the tyrosine 14 and the threonine 14, right? So when there will be a phosphorylation of the threonine 161, right, and when there will be a phosphorylation of tyrosine 15, and then there will be a phosphorylation of, then this phosphorylation of the cyclin CDK cyclin complex, right? So for example, from here, right, so it is actually going to, so then there will be a dephosphorylation, right, and because of this dephosphorylation, only the CDK1 and cyclin complex where the phosphorylation is at the threonine 161, this will be into active form, and that's how the cell will enter into the mitotic phase, and slowly the cyclin B is going to be degraded, and then the CDK1 is actually going to be available. But this CDK1 is still active because 161 is phosphorylated. So then what will happen? There will be a dephosphorylation, and the phosphate group from the 161 is going to be removed. And then there will be a reassociation of the new molecule of cyclin, and that's how it is going to form this complex, and then there will be a phosphorylation. Because of that, there will be a phosphorylation of these three molecules. Then we have the different mode of CDK regulations. So you can actually have the CDK regulation by three different modes. One is with the association of the cyclins. Then you also can have the association with the CDK inhibitors, right, or CKIs. Then you can also have the inhibitory phosphorylation of the threonine 14 and the tyrosine 15, and then also going to have the activation by the phosphorylation of the threonine 161. So you basically can have the different mode of CDK regulations, one with the protein interactions, the second is the phosphorylation. So if there will be a phosphorylation of 161, then it will be actually going to activate the TDKs. If there will be a phosphorylation of the threonine 14 or the tyrosine 15, then there will be a inhibitory actually. So now what we have understood? We have understood that the cell is dividing when there is a need, right? Like for example, we have taken an example of injury or to replace the old cells. Do you know how these induce this process? So when there is a loss of cell, the body secretes some signaling factors such as growth factor, which induce cells to enter into the cell cycle to replace the lost cells, right? And these are the some of the growth factors like the EPDGF, FGF, PDGF, TGF beta, and GF are some of the growth factors. So growth factor, what these growth factors are doing? Growth factors are stimulating the various signaling pathways such as RAS, MEK, ERK to induce the expression of cyclin D1. So they are actually inducing the expression of cyclins, different types of cyclins, cyclin D1, then you have the cyclin B and all that, right? And then CDK4/6 then complex with the cyclin D1 and then allow the cells to the pass through the quiescent stage, that is the G0, and restriction point to enter into the G1 phase. Cyclin D1 is readily degraded in the absence of the growth factor to check onto the cell division. Initially, we have seen that the cell division may also cause cancer, but under normal conditions such thing doesn't happen because there is some cellular protein which play important role to prevent the progression of the controlled cell division to the uncontrolled cell division. So it is crucial that the cell must not enter into the mitosis until the replication of the genome has been completed. So until the S phase is not been done, the cell will not enter into the M phase. Several cell cycle checkpoints, DNA damage checkpoints function to make sure that the DNA damage is not replicated and will not pass on to the daughter cells. This means it actually ensures that there should be no mutated DNA, there will be no damaged DNA should be passed on to the daughter cell, so that the daughter cell is actually going to replicate or going to harbor these damaged nucleic acid from the parents, and that's why it is actually going to have the higher probability that they will actually develop into the uncontrolled cell division, and uncontrolled cell division will lead to the production of the cancer cells. There are also sense the damage or the incomplete replication DNA to prevent the cell cycle progression before the DNA gets repaired or replication get completed. DNA damage checkpoints such as G1, S and G2 phases of the cell cycle. Failure of the DNA damage checkpoint will lead to the accumulation of the mutation in the progeny of the cells and increase the probability of the developing the cancers. How does the checkpoint on the DNA damage and unchecked, unreplicated DNA? So how the this kind of methods are ensured within the cell or will be present in the within the cell, that which will keep the cells under the unproliferated state, or will actually keep the cell cycle within the check. So cell cycle arrest upon DNA damage. So if there will be a DNA damage, this DNA damage could be happen because of many different types of events. All these we are going to discuss when we are going to discuss about the DNA damage and the repair mechanisms. So you are actually going to have the two important protein. One is called ATM, the other one is called as ATR, and there are two crucial protein of the which play actually important role into the cell cycle arrest upon sensing the DNA damage, because it is very important that you should have the complete replication of the DNA. If there will be a DNA damage, that portion will not be replicated, and that's how the daughter cell will not have the information of that damaged DNA. So if you have a damaged DNA, if the parents are actually having the damaged DNA, better to first take care of that damaged DNA, first replicate that damaged DNA, repair the damaged DNA, and then you go for the cell division. So basically these proteins, the ATM or the ATRs, are ensuring that the DNA damage should be sensed, should be repaired, and then you go for the cell division. So ATM and the ATRs are the protein kinases which then activated, there are unreplicated or the damaged DNA, right? When there are any double-stranded breaks in DNA, ATM is getting activated and phosphorylate the CCK2 protein or the checkpoint kinase. Whereas upon unreplicated DNA, ATR gets activated and phosphorylate the CHK1 protein kinase. CHK2 and CHK1 phosphorylate CDK25, CDC25 and CDC25C. Upon phosphorylation, the phosphate activities of both the protein get inhibited. As we have seen before, both the phosphatases are required for the activation of the CDK2 and CDK1, and they are involved in removing the inhibitory phosphate group from the CDKs. Its inhibition leads to the cell cycle arrest at G1 to S phase or the G2 phase, and because of that it is actually going to help the cell to get enough time for repairing the damaged DNA, or it will give the enough time for the cell to complete the DNA duplications, and that's how it is going to help the daughter cells to have the complete information of the genetic information what is present into their parents. So there are two different proteins which are very participate into this, right? So RB protein is a tumor suppressor, right, which plays a crucial role into the negative feedback mechanism of cell division and often mutated into the different types of cancer types. RB check the DNA damage before the cell proceed into the S phase to prevent cell division. In humans, the RB is encoded by the RB1 gene on the chromosome 13. Mutation in both the allele of the RB gene in retinal cells leads to the uncontrolled division of the cells and development of the retinoblastoma, hence the name of RB is being given. The activity of RB protein is regulated by the changes in the phosphorylation state. RB binds to the member of E2F family of transcription factor, which regulates the expression of the several genes involved into the cell cycle progressions. In unphosphorylated state, RB suppresses the activity of E2F transcription factor, and hence the gene expression mediated by the E2F is going to be regulated. The phosphorylation of the RB by the CDK4, 6 and cyclin D complex results the dissociation of the RB from the E2F, and hence the activation of the gene expression mediated by the E2B transcription factor. So RB or the retinoblastoma protein is very important, because this is the protein what is being isolated from the eyes and which is responsible for the development of the cancer, and that's how the name of the RB is given to this particular protein. And what the RB protein is doing, it is actually ensuring that there is no damaged DNA being passed on to the daughter cells, and when there will be a mutation into the RB protein, then they cannot actually be able to function properly, and that's how they are actually being responsible for allowing the uncontrolled division of the cells, irrespective of whether the DNA is damaged or DNA replication is not being complete. And that's how it is actually going to allow the production or the development of the different types of cancers. Then we have the p53 proteins. So p53 is another mutated protein in cancer and commonly known as the guardian of the gene. In mammalian cells, the cell cycle arrest upon DNA damage is also mediated by the action of the protein p53. P53 is another tumor suppressor gene which gets phosphorylated by both the ATM and the checkpoint 2 in the response of the DNA damage. Upon phosphorylation, the p53 gets stabilized, which otherwise rapidly degraded, resulting into the increased p53 level. The p53 is a transcription factor, and increase of its level, the induction of the expression of CDK2 inhibitor p52, which inhibits the complex of CDK cyclin E or the cyclin A. Inhibition of the CDK cyclin complex arrests the cell cycle at G1 phase, and the inhibition of CDK2 cyclin A complex arrests the cell cycle at the S phase. So basically, when there will be a double stranded DNA break, the ATM is actually going to recognize these breaks, and that's how they are actually going to phosphorylate the p53. And there will be, when the p53 is getting phosphorylated, then it is actually going to bind the damaged DNA, and that's how it is actually going to increase the level of the CDK inhibitor P21 or which inhibits the complex of CDK2 cyclin E or cyclin A, and that's how it is actually going to arrest the cell cycle at the G1 phase or the S phase, and that's how it is actually going to allow the cells to repair these double stranded breaks, and that's how it's actually going to ensure that the daughter cell should receive the complete genomic DNA from the parent cells. So this is all about what we have discussed in terms of the cell division and the growth, what are the different types of events are happening, and what are the different types of way in which the prokaryotic system or the eukaryotic system is regulating the cell cycle. And what we have discussed that the cell cycle is a very complex event, right? It requires the cell growth, it requires the duplication of the chromosomes, it requires the distribution of the chromosomes, and lastly it requires the cell division, right? And all these four fundamental events are occurring differently into the prokaryotic system or the eukaryotic system. Even the machinery with what is required for regulating these events are also very different. But the checkpoints, the purpose of the checkpoints and the purpose of regulatory protein is that it should ensure that DNA is being replicated completely from the parents, so that the complete genomic information should be passed down to the daughter cells. And in this context, we have discussed about the CDKs and cyclins, we have discussed about the DNA repair and the checkpoints, and we have discussed about the role of the RB protein as well as the p53. So with this, I would like to conclude my lecture here. In our subsequent lecture, we will discuss some more aspects related to cell division. Thank you.